Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Epen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@epen.info. My guest today is Professor Eleftherios Mylonakis, who is a professor of infectious diseases at Brown University and the chief of infectious diseases at Rhode Island and Miriam hospitals. He is also the director of the COBRE Center for Antimicrobial Resistance and Therapeutic Discovery. He is assistant dean for outpatient investigations and director of the Center for Outpatient and Longitudinal Medical Research at the Alpert Medical School of Brown University, and professor of molecular microbiology and immunology. He has 8 patents and almost 400 articles in peer-reviewed literature. Welcome, Eleftherios.
Hi, Gil. Nice to be with you.
I want to start with some of the very interesting research that's going on in your lab at Brown, especially in the area of surrogate invertebrate hosts for antimicrobial research. And you say that compartmentalization of pathogenesis-related research into an analysis of the pathogen, the host, or the antimicrobial compound has largely been dictated by the lack of model systems in which all of these approaches can be used simultaneously, and by the traditional view that microbiology, immunology, and clinical biology, as well as pharmacology, are separate disciplines. You say that we have, gene-based tools now to sort of integrate all of this and accelerate discovery and development. Could you talk a little bit about that?
Yes. Well, I think that this is part of a challenge, and is also recognizing how my brain works better.
Yes.
So it's a excuse probably for me to just allow my brain to work on different parts, so I don't get bored.
Right.
So I sometimes am amazed by people who can spend decades or their whole careers studying one protein or one part of one particular host pathogen interaction.
Yeah.
And my brain works a little different. I try to stay stimulated. So it sounds good, but this is a vote against the Aristotelian approach of making boxes and trying to differentiate disciplines.
Yeah.
And my approach is more towards trying to put a problem and try to find the best way to get an answer about this problem. So instead of using trying to find the best use for my hammer, I just pose a question, and then I see what scientific approach I can use in order to get closer to an answer to this question. So So this is the general frame of mind, and then I'll talk to you about the invertebrate model hosts. So I cannot really take a lot of credit about that. This was developed in the lab of my mentor, former mentor and current colleague and good friend, Fred Ausubel, up at Harvard, who allowed me to join his lab as a postdoctoral fellow. And he developed the approach of studying small hosts. He started with C. elegans.
Yeah.
C. elegans is a small nematode, 1 millimeter in size.
These are roundworms?
This is right. Yes, correct. Roundworms. The first animal that had its genome sequenced, the model host, but it's a very small host. They live in the environment. We believe that they survive by eating microbes.
Yeah.
And they have a very rudimentary, relative to the human, probably rudimentary host response system. But the important thing is that if you have a small micro-infection in this model host, then you don't have to do an in vitro assay.
Yeah.
This is a host that is big enough to allow you to study host pathogen interaction without having to use mice or do expensive. You still can do high throughput screening of thousands and thousands of compounds.
Yeah.
So you can have in a small well, in a 384 well plate, you can have a pathogen, you can have a host, and you can have a compound, which breaks through the host pathogen interaction, or for me as a physician also adds the practical aspect of how can I treat this infection.
Right. So this is high throughput screening. So just very simplistically, you have thousands of small wells where you have these worms, you have the pathogen in there, and then you're attempting many different compounds to see where the activity might be.
Yes, that is a very simple way. That's how I explain it to my daughters, and that doesn't sound very novel, but it is quite interesting actually, because the way that we do it is screening by imaging. So we can screen thousands of compounds every week, and also it's interesting is that we use also a robot to dispense the worms,
right?
And that allows a very high, enables us to be high throughput, have a standard number of worms of exactly the same size in each well. So we start with the same number, and then we can use a fluorescent dye to stain the dead worms. And then we do screening by imaging in order to identify the wells where there was a lot of survival. Yeah. And then we can go and identify the compound that was there. And what is fascinating with that is that you identify compounds that you wouldn't have been able to find with an in vitro screen.
Right.
And also you can find compounds that they work on the immunity of the worm. So they augment the immunity and allow the worm to fight the infection. Or you have compounds that decrease the virulence of the pathogen. Those groups of compounds you cannot really find with a straightforward in vitro screen.
Right. Right. And so, in terms of the different preclinical testing that we typically go through in pharmaceuticals, this has a potential of really going to the clinic a lot faster, I would imagine, right?
Well, again, I have to take you 2 steps back in order to answer this question. So, if you allow me,
sure.
This is not done by choice. For the longest time, the drug discovery was mostly left in pharmaceutical companies, where they had their own libraries, they had their own screening equipment, they had their own medicinal chemists, and they were able to go through and discover new antimicrobials reasonably, with some reasonable time frame and dependable approach. So the problem is that the pharmaceutical companies abandoned this field for many reasons. I don't know if you want to discuss them, but
Sure, yeah.
it's not part of the particular question. But in any case, they abandoned it, so it is now left to academia to try and fill that gap. And academia can do some of the aspects of pharmaceutical drug discovery, but not as effectively. And we have to find shortcuts. Right. So there are a couple of areas where academia has great difficulty. The first one is the identifying a hit. Yeah. So for academia, we need to be able to identify usable hits more effectively than pharma, because for us, following up on a hit means a big commitment of time. We have a small research lab. We don't have the big number of people that can follow on hits, and we don't have the capabilities on and the technology that is needed. So we need to rationalize our hits and have a reasonable low hit number. Right. The second part is we need to screen for toxicity really quickly. Toxicity is the bottleneck of antimicrobial drug discovery. Yeah. As you have seen with the discussion about COVID-19, finding compounds that in vitro have some efficacy is not very difficult. Right. The problem is that some of them are not good drugs. They are more like things that we can use for cleaning or things that are very toxic. Yeah. So that is another bottleneck that we need to address very quickly. So we don't waste our time with toxic compounds that take us nowhere. So if you have a host in there, a eukaryotic cell, if you have something that is very toxic, then it will kill the host. It won't come as a hit. You don't waste your time trying to discover it. Yeah. Finally, one other area where academia has great difficulty is developing analogs of compounds. Mm-hmm. So when you go from a hit compound in order to get to a lead compound, which is a starting point from going to mouse studies, etc., you need to take your chemical and optimize it. Optimize usually means improve efficacy, decrease toxicity. That is the easy thing. Yeah. Pharmaceutical companies used to be able to make hundreds of those analogs. As soon as they had a hit, they hundreds of analogs, and then work from there, find the part of the molecule that is the most active, and improve on that. Right. Now, even if I have 2 or 3 collaborators, medicinal chemists that collaborate with me, it's impossible to get hundreds. At the most, we can convince them to get a handful of analogs. Right. So in order for us to get those targeted analogs, we need to have some in vivo results. That also is an added value on doing the screening, the high throughput screening using a host like C. elegans. So hopefully I went a very circular way to answer your answer, but I think that I hope that I gave you enough context.
Yeah. So 2 things. One is, this type of an approach is more in discovery where you're doing the screening. And since you say it's in vivo testing, you have less chance of a toxic compound entering the preclinical development phase. Do you see opportunities there to compress the timeline too in preclinical? So you know, both finding the compound as well as when you find it, because it's an in vivo experiment, you are likely finding things that are not toxic, right? So you don't necessarily have to go through, the elaborate process pharma companies go through in preclinical.
Well, we do screen for toxicity. That is, of course, you have to screen for toxicity against mammalian cells, lines, but you just increase the likelihood that your hit will make it through those toxicity screens. Yeah. So that is, there is more than 50% of our hits have no toxicity in mammalian cell lines. I would say that is higher, significantly higher than 50%, which is extraordinary. Yeah. Then then I have to say that our main goal is not only to find the compound. Our main goal is to find the target.
Right.
The mode of action is what is academically more stimulating. And also it gives us the flexibility. If this compound turns out to have a very poor pharmacokinetics, or this compound has some unexpected toxicity or some other difficulty, if we find the target, then we can allow our lab or other labs to screen against the target.
Right.
So if we identify a novel target for a druggable compound, then even if that particular hit doesn't develop and go to preclinical steps, still that is a target where others can screen their compounds,
right,
and they can identify new compounds that are less toxic, and they can move those forward.
Right, right. Yeah, so when you say, Eleftherios, when you say pharma has abandoned this, do you mean pharma has, is that specific to antimicrobial research, or you're saying in general, high throughput screening type approaches in discovery, pharma's not doing too much of that anymore?
I don't know about, in general, the number of high throughput screens that they do for other fields.
Okay.
I am working on microbial drug discovery, and that is a particular problem. I'm sure that they have had great progress on discovering new medications, for example, for cancer. I was just I was renewing my boards for medicine last year, and there were, since the previous time, 10 years ago, that I had to go through the same process, there were more than 200 new antineoplastic agents that I had to learn. Mm-hmm. And from antibiotics, it was less than 10.
Yeah.
I was so jealous of people who work on oncology, all those big discoveries that have happened there. So there are many reasons. This is not meant to be necessarily putting the blame on pharmaceutical companies. This is a very distorted market that needs to change. But I'm stating as a matter of fact that antimicrobial drug discovery has to evolve, and the market has to evolve in order to find new treatments. Otherwise, we're going to be seeing more and more resistant infections that we cannot treat.
Yeah, so I think, I haven't followed the field that closely, but, economics to some extent drove big pharma out of antibacterial research, perhaps in 2000s, late 1990s. And that gap, as you say, was taken up by academic organizations. But increasingly, I guess we are finding we are up against some really resistant organisms, right? And that's creating a lot of problems.
Yes. Before COVID-19, there were projections from WHO was that by 2050, the infections from resistant pathogen would overtake cancer as a cause of mortality. So, and no one really knows how the situation is going to be post the pandemic and COVID-19 and people in the ICUs intubated for a long period of time. They're very resistant Acinetobacters. They're very resistant Klebsiellas, of course, ESBLs, and even more resistant than that. And MRSA now, it's part of the hospital consult service. Sometimes I have more patients with MRSA than anything else on my list of patients to see.
Well, yeah.
So those are very severe infections. Those are very aggressive pathogens, and we, for the longest time, we used to believe that those would be only associated with hospital patients, and that they wouldn't never come from the community, and that was completely false. We have people, young women who sometimes come from the community with UTI that is caused by an ESBL, very, very resistant gram-negative.
Yeah.
And they had never been to the hospital before. And regarding your point about the pharma abandoning over the last, 1 or 2 decades,
Right.
abandoning this field, even before that, there was a lot of Me Too kind of molecules.
Yeah.
There are two periods of decreased discovery. It was one period where everybody was trying to get a new cephalosporin, third generation cephalosporin, like a Me Too kind of approach. And then there was a second period where the whole project was abandoned. And we can go into why this happened. Again, I don't want this to be like an academician blaming pharma.
We can go into the MRSA discussion, but I want to also ask you: is a surrogate host approach what for viruses too?
I'm not a virologist, so we do know that there is there is some work on using some hosts and in virology. For me, pathogens are two kinds of pathogens.
Yeah.
The pathogens that go from human to human, and viruses are, usually the most common pathogen that goes under this category.
Yeah.
And there are pathogens that go from that they can live in the environment. Right. So those are the two categories, and I'm good at this second category. Those are bacteria, fungi, whatever can live in the environment, because that's where invertebrate models and those pathogens develop their virulence in order not to kill humans.
Yeah.
Humans became a significant part of the biomass just recently. Those pathogens develop their virulence in order to live in the environment, avoid amoebae or survive amoebae, invertebrate hosts, insects. That's why they develop their virulence. And that virulence happens to translate, in some cases, in human disease. So if we go back and we take an invertebrate host, actually what we do is not we are discovering something new. We go back and we try to see the host pathogen interaction where it's supposed to happen.
Yeah.
In a more environmentally relevant host. Now, going back to your answer, the viruses and those that go from human to human, I'm not very good at, so I will let others discover those.
Yeah, so there's a diversity of a diverse set of hosts there, right? So I know that amoeba, even zebrafish have been tried, right? So there appears to be a lot of flexibility there in terms of attempting different things.
Yes. What we have a meeting of model hosts every couple of years. It takes place in Greece, in a Greek island every time, usually Crete, but other islands as well. So we are very happy to go to my motherland and discuss about model hosts.
Yeah.
And every time that when I take the plane to come back, I realize that there is not one single perfect model host. Not even the humans are a good model host, quote unquote. I often say the example of fluoroquinolone that was developed by Pfizer, and went through all the phases and became widely available and FDA approved, and it would kill gram-positives, gram-negatives, and anaerobes. I remember a good friend of mine called me and asked why infectious diseases physicians are needed anymore, because he would give Trovan to everybody and everybody would be fine. Yeah. And then it was discovered that it causes liver disease and liver toxicity, and it was pulled from the market.
Right.
So even large clinical trials in humans have their shortcomings. So as a result of that, you can imagine how hosts, mice, rats, worms, insects, zebrafish, amoebae, they can all add something useful.
Yeah.
They can all answer some question, but you have to put everything into a context. And what we're trying to teach the younger scientists in the field is try to understand what the host can help you discover and understand its limitations, no matter what the host might be.
Right. Yeah, I mean, Provenus, as you know, is a famous, very broad spectrum antibiotic that Pfizer tried, which was pretty successful, except it had so many scripts in very short period of time that, you know, the toxicity that typically shows itself over a longer period of time actually showed up in 6 months.
Yeah.
But I think that the drug is still there as a hospital-administered drug, I believe. I'm not sure.
I haven't used it for 22 years, somewhere there.
Okay.
23 years,
Okay.
yeah. So it could be theoretically available, but I don't think that you can really use it.
Okay.
And that is an example of how, you know, as I said before, not even the human clinical trials can be 100%.
Right. Yeah. So I want to talk a little bit about it. So in one of the recent papers that you have, you say that there is a critical lack of therapeutic agents to treat infections caused by non-growing persister forms of methicillin-resistant MRSA. And you have some ideas there to go after this. Could you talk a little bit about that paper?
Yeah. We that was again serendipity took us that way, and just followed some of the leads and tried to understand what we were seeing. It took us through some interesting, what we think that is very interesting areas of discovery. So Staphylococcus aureus has a very unique feature that is it can go into a dormant state and stay there metabolically inactive for a long period of time. This is quite interesting because it could explain why I need to treat people who have staphylococcal infections for a longer period of time, and why the relapse rate is higher than in other pathogens.
Right.
Essentially, every single staphylococcal cell, bacteria, can go into this dormant state, and makes it very difficult for the drugs to kill it, because most of the drugs, they need to get into the cell, find something that has to do with something that with some function that is important for the cell to survive and really work through that route by disrupting that important function.
Yeah.
When the cell, the bacteria, the bacterium is metabolically inactive, then they cannot really penetrate. There is way less that they can do in order to kill the bacterium.
Right.
Now, the only one of the few parts that the bacteria cannot do much about is the bacterial membrane, the outer layer of their existence. The part that makes the bacteria differentiate from the environment.
Right.
That is difficult to hide, even if you are a very smart bacteria like Staphylococci.
Yeah.
So the problem with the membrane is that the eukaryotic cells, the human cells, also have membranes. So for the longest time, we have had difficulty finding compounds that can disrupt and work there, because again, the problem of toxicity. would make this kind of discovery both difficult as well as high risk.
Right.
And interestingly, with a group of compounds that we found through our high throughput screens, we were able to believe in the lack of toxicity because of the C. elegans story that I told you before.
Yeah.
And we found groups of chemicals that really affect parts of the cell membrane of Staphylococcus. And now we are trying to discover what exactly they do in order to disrupt the membrane and find the exact targets of those compounds on the bacterial membrane. But this is quite exciting because those chemicals, alone or in combination with existing antibiotics, they can disrupt the cells and kill bacteria even when they get into that dormant state that I described before.
Right. Will they be slow acting, Eleftherios? If once it's in production, will it be sort of a chronic administration?
Can you clarify your question?
Will it be sort of you have to treat for a longer period of time to make it effective?
Yeah, for the regular antibiotics, I have to essentially outweigh the Staphylococcus.
Yeah.
In order to get rid of a staphylococcal infection, I have to give my antibiotics for a long period of time that is too long for the bacteria to stay in that dormant state, and then I can kill them. The hope is that with these more aggressive compounds that work against metabolically inactive staphylococci,
yeah.
I can just give them for a short period of time and can go and disrupt the membrane, and I won't have to outweigh the staphylococcus for it to get out of that dormant state in order to kill it.
Okay. It's sort of cleaning the system, so to speak. They're not active. So these agents, they're not very toxic, but they can actually disrupt the membrane. They can selectively disrupt the membrane of the persister, MRSA.
Yes, we feel that they have some. We don't feel, our data indicate that they have some selectivity.
Yeah.
For one reason or another, and we're still trying to, in a molecular level, they have some selectivity towards the bacterial membranes and not towards the mammalian membranes. So that makes them druggable, i.e. not very toxic.
Right. Yeah, I want to touch on another paper that you have entitled A Study on the Cost Effectiveness of New Diagnostics and How They Can Be Implemented, where you show statistically that combining molecular rapid diagnostic tests with antimicrobial stewardship programs seem to have reinforcing benefits. Could you talk a little bit about that?
Yeah, well, this is I grew up in a modest household where we had to balance the books on a monthly basis.
Right.
So there are two ways to balance the books: is to get more money coming in and to decrease the amount of spending that you do.
Yeah.
So increase the money coming in. In our case, is increase the compounds that you have available, the antimicrobials that you have available to use. Decrease the spending is use your existing antibiotics strategically, so you can maximize their use. So this goes to the other part of the equation, how to be very selective and appropriate in the use of antibiotics.
Mm-hmm.
And there are two areas where we use a lot of antibiotics. The first part is what we call empiric therapy. Yep. Empiric therapy means the patient has an infection or looks like the patient has an infection, and I want to cover everything. I don't know what's going on.
Right.
Infection can be in the belly, can be in the lungs, can be in the blood, It can be a urinary tract infection, can be a gram-positive, a gram-negative, can be even some fungal infections. Some fungal infection can be a virus. Throw everything that they have. Use 2, 3, sometimes even 4 antimicrobials at the same time. And that can go for some time because our diagnostic ability is completely archaic.
Right. Yeah.
We talked about the antibiotics that have limited options, and we have had a delay in significant discovery for a few decades now.
Yeah.
Most of our diagnostics, especially for bacterial infections, depends still on the waiting for the bacteria or the fungus to grow.
Yeah.
We're waiting for it to grow. And this makes no sense right now because we live in an area of molecular science.
Right.
But the clinical part, because it is always discoveries take a long period of time to go from research to clinic, still depends on blood culture and urine culture and susceptibility testing. And there is a growing interest in trying to use molecular diagnostics for rapid testing.
Yeah.
I remember vividly a few years ago when I had a patient who was septic, very ill, in the ICU, was on 4 different antimicrobials, and I would go to the micro lab and ask if they had any results so I can optimize therapy, stop the antibiotics that don't work, give the best antibiotic that I have for the infection that they discovered. They would tell me, nothing grew. At the same time, for my own research lab, I had a whole genome sequencing of different enterococci, and I was able to get it back in 5 days.
Right.
Whole genome sequencing for a series of enterococci.
Yeah.
And it made no sense because in the one case there was a human who was very ill and sepsis means the chances for survival go down by the hour.
Yeah.
On the other hand, I had a research project that can wait. So you understand that there is a real need for new molecular diagnostics. And the problem that we have there is how to convince the hospitals to invest. Healthcare, although we spent $1 out of $5 in this country, even before the pandemic, was spent in healthcare, still the margin that most hospitals and healthcare networks work with is very thin.
Yeah.
So in order to have a new approach, a new diagnostic implemented, you need to make the argument that this will save money.
Right.
There is fragmentation of payers and other things that we can discuss, but at least in order to get the decision makers to listen to the need for this new diagnostic and new approach, you need to make the argument that this will save money, at least overall.
Yeah, so the score diagnostic tests are not sensitive, specific, and fast enough that we tend to use antibiotics, sort of in a cocktail fashion. So we are overusing antibiotics because we just don't know precisely what the infection is and where the infection is, right?
That's exactly right. And there is a lot of toxicity added. Yeah. And as a physician, this is what I am concerned the most. And also, the problem doesn't stop there. If you have a patient, even usually you get the results in 3-4 days, a blood culture will turn positive or some other result will come back. And by that time, the patient usually, very frequently starts to improve with this cocktail of antibiotics.
Yeah.
Then clinicians are a little concerned. They say, why stop the antibiotic now? The patient was dying, and now the patient is getting better. Why? Let's keep them going, all of them. So you hit the point. If you don't do it, if you don't change the treatment really, and you don't impact the treatment really at the beginning, and you don't have good diagnostics, even if you get the information finally, sometimes that information does not translate into clinical action.
Yeah.
For a good reason. So what has happened? We have this tool that is called antimicrobial stewardship. Mm-hmm. This is a wonderful tool in the reasonable use of antimicrobials in the hospital. Mm-hmm. This is usually in infectious diseases with a pharmacist collaboration through the administration, where they try to help. Essentially, what they do is try to educate physicians to think about the overall picture. Yeah. If physicians, we always think about our particular patient that we have in front of us. We feel the need to advocate for this patient, do the best that we can in order to help this particular patient. And sometimes that means that I have to give 3 or 4 antibiotics, give all the antibiotics, give broad spectrum antibiotics, and try to do my best to help this particular human being. Right. Now, the antimicrobial stewardship comes and tells me, wait a minute. Think about all the patients who come to the hospital. Mm. If you breed the resistance with the inappropriate use of an antimicrobial, then that will kill, maybe won't kill your patient right here, but might kill this particular resistant pathogen, a patient who is not even in the hospital right now. Mm. Mm. If you develop a resistant clone of a gram-negative, and that lives in your ICU or lives in your burn unit or surgical ICU or trauma ICU or transplant ward, then that can kill somebody who is not even in the hospital right now. So the antimicrobial stewardship tries to rationalize and help us, educate us to think about the big picture. And that tool we feel is the optimal tool in order to take that information from the new diagnostic tools and put it into clinical action. Yeah. Because it's there, it exists, it is widely accepted. Now it's part of every hospital system, and we soon believe that it's going to be part of every long-term care facility and even the community, and can take the information from the micro department, from the new diagnostics, and implement it to make a difference.
Yeah, but it's also a worldwide problem, right? So to have a systemic effect, it has to be implemented, I would imagine, across the world.
Yeah, well, that is ideal, but as we're learning now, even small wins count.
Okay. Yeah.
I think that sometimes trying to get everything in place in an optimal way, it's results in inaction. Yeah. We believe that get the antimicrobial stewardship as far as it can go, and then fight it to go even further. Yes, we need to have antimicrobial stewardship throughout healthcare, throughout countries. For example, we found that athletes, collegial athletes, have a high incidence of MRSA and other resistant bacteria. Right. We found that travel to some countries is associated with colonization with resistant bacteria. Mm-hmm. Yes, we need to have stewardship and infection prevention as we understand it now with COVID-19. It has to permeate our society. We are guests in an environment of microbes. Right. Then we need to have those filters. However, we have to act as we work on getting this through. the whole system. And right now, antimicrobial stewardship acts very effectively in the hospital. Let's empower it. Let's strengthen it. Let's have it use all the tools and be our advocate for the optimal use of new diagnostics that, in result, will decrease cost, improve outcomes, and decrease resistance.
Yeah, I find it fascinating, Eleftherios. So you said the prevalence of MRSA colonization among asymptomatic athletes is comparable to that among individuals with chronic illness, and possibly higher among them, even twice that for patients in intensive care units. So I don't know if this translates into the COVID issue that we currently have. Asymptomatic patients may be carrying a higher level of or higher load. I don't know if it translates. I'm just asking if that is possible.
I think that there are a couple of things here.
Yeah,
more than a couple. Let's take it one layer at a time. First of all, you said that indeed, it's not my thinking; it's what the numbers say.
Yeah.
Indeed, collegial athletes and other athletes have a higher incidence of MRSA colonization than what we see in patients in our ICUs.
Right.
That is an unbelievable thought process. There is every so often you hear about a football team or some other team that has an MRSA colonization. There are a couple of extraordinary good professional basketball players who had their career cut short because of MRSA infection. And we have athletes, we have young collegial athletes who come to the hospital and their first admission is with MRSA infection that they didn't even know that they are colonized with.
Yeah.
So this is something very, very important to keep in mind, because as humans, we tend to believe that those things are somebody else's problems. No, it's our problems. We all have bacteria. We are all vulnerable, and in that regard, this aspect can translate to COVID-19.
Yeah.
The aspect that we usually, our first reaction is first or second reaction is, of course, as part of the denial mechanism to coping with a new reality, is to say, this doesn't affect me.
Right.
And we need to realize that we are all together on this.
Yeah.
It can be COVID-19 today, it can be resistant bacteria tomorrow, it can be something else. What happens? It was Ebola a few years ago, and HIV even more few decades ago. Always we try to think that this is not a problem that could impact us and our family members, and we are always wrong, because infectious diseases is it's always a public health issue in its core. Right. And we're all public and we are all at risk.
Right. Yeah, that's an important point. So in conclusion, Eleftherios, looking at all your research at Brown and elsewhere, if you look forward 5 years, where do you see the maximum potential? So, research around the surrogate host, high throughput screening that you are doing, the diagnostics, fast diagnostics, coupled with other type of programs, and this colonization of microbial agents, in a group of people rather than a single individual. Where do you see the maximum potential in terms of making an impact?
Yeah, the maximum potential, in my mind, is with new diagnostics.
Yeah,
this is something that it's a long time coming, and there are good discoveries. There is excellent, exceptional, good techniques, and there is no question that those will change the way that we diagnose diseases. There is already a trend towards new diagnostics. We have new diagnostics for pulmonary infections and CSF processing. This is the cerebrospinal fluid. I think that it's a matter of time that those will change completely.
Yeah.
What I say to our students is that 10 years from now, they are going to tell their students about the need to wait for bacteria to grow, and they will think that was crazy.
Right.
In the same way that when I start my presentation and my discussion is before the antiretroviral era of HIV, and the students think that I'm just too old. And clearly that is the one area. Then an area that I'm reasonably optimistic is antimicrobial stewardship. I think that by necessity, we are going to have to optimize the use of existing antibiotics.
Yeah.
The area that I'm less enthusiastic about and less optimistic about is the area of new antimicrobial drug discovery. Because there you need to reframe the market, you need to remake the whole field without the existence of big pharma, at least for the part of the very early antimicrobial drug discovery. So in that regard, there I'm less optimistic about.
Yeah, it looks like diagnostics is foundational. So if you get fast, specific, sensitive diagnostic techniques, that should result in more optimum use of antimicrobial agents and generally perhaps slow down this path that we are on in terms of resistance, right?
That is exactly right. I told you we discussed about the empiric therapy, which is therapy when I don't know what exactly is going on with my patient and I want to cover everything. The other part where you use antibiotics is we don't know how long to use them. And hopefully the new diagnostic will allow me to use it for a specific and personalize my use of antibiotics.
Yes.
Personalized means using the antibiotic for enough time, but not for too long. Because right now, for particular infections, they ask me how long to give. And I say, for example, 2 weeks, 3 weeks.
Yeah.
But this is an extreme. Some people might need a little longer than that. Some people might need just a week.
Right.
I don't know. I don't have a tool to help me personalize and tailor-make the duration of antibiotics. If I have a very good diagnostic, then I can probably screen exactly for the existence of any dormant bacteria, any small amounts of bacteria, and if I get a negative signal, stop the antibiotic and follow. That will also decrease the overall use of antibiotics without putting the particular patient at risk.
Yeah. Some of these techniques being developed in artificial intelligence, for example, could be useful too. In terms of, as you say, personalized medicine. Really identify what is precisely needed for that individual rather than using macro heuristics, right?
Yes. We like large data. We like artificial intelligence. But when we go down to the particular human being, what I need to know is, are there any more staphylococci in the bloodstream? Are there any more staphylococci in that joint that will need me to extend the antibiotics? So that is the challenge that I have in order to stop the antibiotic and not get it go too long.
Yeah. So that is more in the area of diagnostics. So diagnostics, it sounds to me, has to improve significantly.
Mm-hmm. Yes.
So. Yeah, this has been excellent, Eleftherios. I really appreciate the time that you spend with me, and good luck with all the work that you're doing at Brown.
Thank you very much, Gil. It was really stimulating talking to you, and thank you very much for giving me the opportunity to reflect on those works.
Thanks very much.
Bye.
Bye.